Bismillah ar-Rahman ar-Rahim. Today we will start the second part of the course entitled Eradication of External Parasite. We already speak about the harms of ectoparasite on their host and the types of ectoparasite. We start with the ticks. First, we take the life cycle of ticks and how we can put eradication program of tick. We start with the physical control program and then chemical control program and then with the integrated control program. The integrated control program involves some steps that we should start the strategic control. Usually we make assessment from time to time, maybe periodic or annual, and then usually the assessment depend on the experimental data and the field result, and usually we should concern about the cost of each step of this plan. Because of that, the integrated control program we needed because of the disadvantages of the chemical control program in resistance, in environmental contamination, and the concern about the effect on the human health and the host health. So a new approach is needed for the natural host safety and environment safety. Usually we depend on the development of the molecular techniques for giving a powerful insight into the diagnosis and then reinterpretation of the results. The integrated control program included vaccination, breeding cattle for tick resistance, endosymbionts, ethno-veterinary practice, and finally the biological control. First, vaccination. Vaccination is the biological process to provide active acquired immunity against ticks. The first attempts of vaccination using a crude vaccine. The crude vaccine used the tick extract of the semi-engorged adult female and inject in the host, so the host will start to build up a specific antibody against the tick. So when another tick come and start to suck the blood, so the antibodies of the ticks will start to act and destroy the cell lining of the tick's gut and allow the blood to escape into the hemocyte. So that result in some ticks die and others reduce fertility of both male and females. Then vaccination start to develop new methods for formation of vaccine. For example, the genetically engineered and subunit vaccine. The antigen used from the tick gut absorptive surface. After injection in the host and the antigen-antibody reaction occur, if another tick suck the blood, there's a prevention of the overproduction and death of the tick. Breeding cattle for tick resistance. It's the development of cattle lines or breeds with enhanced genetically based resistance. For example, the Zebu and Sanga cattle, which are indigenous breeds of Asia and Africa respectively, after their initial exposure to the ticks, they become very resistant. On the contrary, the European breeds, after initial exposure to ticks, they are not used for the tick infestation in the cold countries. So when we bring to and raise in hot countries, they remain susceptible after the initial exposure to ticks. The repellency of the Zebu and Sanga cattle are usually related to the thick movable hide, which covered with a short, straight, non-modulated hair with a high skin vascularity. Also, the well-developed panniculus muscle, which is a sheet of dermal muscle which developed in mammals that allows the movement of the skin independent of the movement of the deeper muscle in case of the biting of the ticks. Also, the sensitive pilocomotor nerve system, which has an efficient erector pili muscle, which, when contracted due to the ticks, causes the hair to stand on end. And finally, the high density of sweat glands, which stimulates the secretion of sebum in the hair, which is a repellent for ticks. The targeting the endosymbionts of ticks. Endosymbionts is the ticks carry some pathogenic microorganism in their bodies. And these microorganisms and ticks are essential for the survival of each other. So when we make targeting of the microorganism, so we can then destroy or affect the development of the ticks indirectly. There are two types of endosymbionts, either primary endosymbionts or secondary endosymbionts. The primary endosymbionts is the microorganism present usually on the ovaries of the ticks and important in the reproduction. So it's important for the survival of the ticks. Another endosymbionts is the secondary endosymbionts, which is of little or minor importance if we target the secondary endosymbionts that are not essential to destroy completely the host. The second endosymbionts usually related to the nutrient production and the environment production of the ticks. So when we make the elimination of the microorganism that could be deleterious for the survival, growth, and development of ticks. Another factor in the integrated control program that we could use is the biological control. The biological control is the use of naturally occurring species of living organisms, either macro or a microorganism, for example, nematode, bacteria, virus, to make a management of the ectoparasite. This led to reduce the density of the ticks, but we should take care of the introduction of the exotic species in the environment of ticks require a deep understanding of the ecology unless carry a higher risk and undesirable effects. The biological control include pathogens as bacteria of the Enterobacteriaceae family called Sedicillapagi. The experimental studies or data showed that it caused mortality in ticks. The mortality in ticks reached 100% if the bacterial suspension was injected directly to the ticks. But if put in a petri dish and immersed in the bacterial solution, the mortality reached 40%. Also, the other survival from the mortality caused 50% reduce in the egg production due to the severe damage to the vaginal epithelium. The biological control, including also protozoa. The two families, namely Sternematidae and Heterorhabdidae, include about 25 species that are known to be pathogenic to insects. Usually, the infective juvenile stage are applied to penetrate the hemocoel of adult female tick enzymatically and mechanically, then cause release of the symbiotic bacteria, and then colonize and kill the host. Another example of the biological control including the predators. Predator is an example of the macroorganism that could kill the ticks. The natural predators of ticks attacking them in the free-living stage of the engorged adult female in the environment, or they can pick the adult stage or larva or nymph on the animal body. For example, the oxpecker and domestic fowl are usually considered one of the most common predators. Also, the ants and spiders are known to be important predators of ticks. The infertile hybrid is an example of the biological control of ticks. When we make mating of two different species of ticks, the mating will produce a fertile females and a sterile males. That usually happened when mating of the Bophelis annulatus and Bophelis microbolus. So, the back-crossing of the fertile female will produce a sterile male and fertile female. After about 3 to 6 generations, they will be completely eradicate the type of ectoparasite or type of ticks present in the place. But the problem of this method is the cost of production of the hybrids. Ethno-veterinary practice. The ethno-veterinary practice is one of the integrated control program for ticks. The ethno-veterinary means ethno is something related to herbal or plants, and veterinary related to an animal. So ethno-veterinary is a plant which affects an animal. The plants or herbs may cause anti-tick insecticidal effect, or cause a growth inhibit of the ticks, or cause anti-molting, or had a repellent activity. For example, in Punjab, it's a place in India, they're using a plant called taramera to control ticks. Usually, they take the powder of the plant and soaked overnight in water, and then in the morning they give orally to the cattle in summer. Usually, in addition to reduce the tick burden infestation, it used as a milk yield booster and have a cooling effect. So it helps also to sustain the heat resistance in summer. Usually the ethno-veterinary practices are depend on the questionnaire of the old people present in a locality. Usually they took the plant and make crushing and soaking in water. Then sometimes they spray on the animal body or just give orally to the animal. Eradication of lice. Two types of lice are known, the sucking louse and the biting louse. The sucking louse usually suck the blood, while the biting louse bite and feed on the skin or feather of the host. Usually lice is a species specific. For example, the lice on horse don't come for sheep, goat, and other animals. The infestation usually done by direct contact from animal to the other. The life cycle include egg, nymph, and adult. The lice spend nearly all the life cycle on the animal body. That means that when we make a control program, we should focus on the treatment of the animal itself. We are not much concerned here about the control program in the environment. The treatment in animal include the chemical application. We can use any method of the chemical application discussed before as powder, pour-ons, dips, or spraying with an insecticide material. Insecticides usually include organophosphate compounds as diazinon or malathion and pyrethrins as deltamethrin, and we can use also the insect growth inhibitor to eradicate lice. Eradication of fleas. Fleas are usually a blood-sucking insect which developed massively in animal house in dry bedding. The life cycle include egg, larva, pupa, and adult. About 95% of the life cycle in present in the environment, not on the animal body, and the life cycle usually takes about 30 days. There is something important here in the life cycle called the pre-emerging adult. The pre-emerging adults usually happen in animal facilities that have not been used for a certain time, and when the animal come back, a sudden outbreak could happen. That may happen due to the pupa can remain in the bedding without hatching for up to 6 months. And when the animals come back, they start to detect their body heat, the movement, the pressure caused by footsteps, and the increased the amount of carbon dioxide from the animal breath, and all that will stimulate the hatching of the pupa to become adult. Prevention and control of fleas. As fleas, the adult individual of the fleas spend about 5% of the life cycle on the animal body, so the animal could be best treated by dusting or pour on. However, the remaining 95% of the life cycle of the immature stage are on the environment. So it's best to direct treatment of the bedding and other resting places, including spraying of one of the organophosphate, carbamate, pyrethroid, or insect-developed inhibitor. Also, elimination of the old building and restricting livestock access to the animal facilities is important in control of fleas. Although a product with ivermectin used for injection is not efficient in the control of fleas, also the vaccination and other biological control is also not effective in the control of fleas. Eradication of mites. Mange mite is a highly contagious disease of livestock animals that live in or on the skin of the host, according to the species of the mite. It's a typical winter pest which favor the humid and cold weather. The life cycle of mites included 5 stages: egg, larva, protonymph, nymph, and adult. There are 4 characteristic species of mites include Demodex folliculorum, Sarcoptic mite, Sauroptic mite, and Choeoptic mite. The transmission within the herd is usually by the physical contact. There is no external vector, and usually the life cycle is on the host, so the eradication program will be mainly on the animal body. The Demodex folliculorum, including the mite, usually present into the hair follicle and species gland that build nodules and papules. So it's not in the skin, but it's on the deep layer of the skin. Another mite which also present in the deep of the skin, which make a burrow in the stratum corneum of the epidermis, parallel to the surface, and deposit the eggs and feces within the burrows. So those 2 types, the Demodex and Sarcoptic, need the control treatment usually involved to reach the deep layer of the skin, not to put topically or to put on the surface of the skin, so the insecticide will not affect the mite infestation. Another species of mite called Sauroptic mite, which is not infectious for human dogs and cats because it's a species specific. Finally, the Choeoptic mite, usually called the leg mite, and also it's not transmitted to human. The predisposing factor of mites, usually in the winter, at which it favors the humid weather. The crowding during the indoor is also important for the spreading of mite, and the stress on the animal usually involved in the infestation of the animal. The preventive measure of mites. As a mange is a contagious disease, so we have to make notification of the appearance of any case of mite, then make isolation of the infected animal and inspection of the herd, avoid overcrowdedness of the animals inside the building, avoid humid hair coat, and avoid stress on the animal. And we should keep the animal in a well-fed and good health and hygienic condition. The exposure of the animal to sun could reduce the humidity and then lower the infestation rate. The treatment of the animal against mite differ according to the type of mite. Control of mites. The control of mites include the skin treatment and injection. The skin treatment could be differ according to the species or the genera of mite involved in the infestation. If it's a demodex or sarcoptic, we need the preparation of the skin before treatment. Usually the preparation include the clipping of the hair, then the remove of the crust after soften with the hard brush soaked in sodium carbonate until the blood oozing, then applying the insecticide. However, if animal in a poor condition or the infestation in a large place of the animal body, it's impossible to treat the animal on skin treatment. So it's better to divide the animal to quarters and start with one quarter and then to another quarter after a period of time. Or it's better to use injection. We could use some of the of the ointment, dipping, spraying, or pouring on after the preparation of the skin, including the sulfur ointment or one of the acaricides. However, we could use injection of the animal using one of the macrolytic lactones, which could give a good protection against mange. When we want to make eradication of mite, we have to put on mind that there is no vaccination to protect cattle from further infestation. There is no repellent to use for mite control. There is no biological control. There is no traps. Thank you.